I think we'll go ahead and get started. So welcome everybody. Today's seminar speaker is our own Dr. David Odde, who is a professor of biomedical engineering here at the University of Minnesota. He's from the Twin Cities and got his undergraduate degree at the University of Minnesota, a Bachelor's of Science in Chemical Engineering, and then went on to earn master's and PhD degrees at Rutgers University in chemical and biochemical engineering. He focuses on mathematical and lab-based modeling of biological phenomenon, many of which are relevant to cancer. He specializes in work on 3D bioprinting, microtubule assembly dynamics, the mitotic spindle assembly, physics of cell signaling, cell migration, especially the influence of cell adhesion and substrate stiffness, which of course is very important for cancer cell invasion and metastasis. He's been the recipient of a number of awards over the years, including an endowed professorship in cell and molecular bioengineering at Michigan Tech, where he started his career as an assistant professor before coming to the University of Minnesota. A career award from the National Science Foundation. He's also been elected to the College of Fellows of the American Institute for Medical and Biological Engineering. Also, a small world thing is that I'm also from the Twin Cities, and my best friend and soccer buddy in junior high was Dave's best friend and soccer buddy in high school. We didn't go to the same high schools, so just a strange overlap. So in Minnesota, it's 1 degree of separation between everybody, I'm convinced. So anyway, he's going to tell you about cancer cell migration mechanics. So, David.
Thanks a lot, Dave. And if it isn't 1 degree of separation, it's just 1 degree.
Yeah, right.
Or both. All right. Well, it's a real pleasure and honor to be here today to talk to you about some of the work that we've been doing in the area of cancer cell migration mechanics. As Dave mentioned, my background is as a chemical engineer. So in chemical engineering, we're interested in how we can describe processes in terms of their fundamental thermodynamics, kinetics, mechanics, and transport. And along the way, I got very interested in that, not in the context so much of oil refining and things like that, but in the context of biological processes, and try to apply the same framework, the same theoretical framework that we use in chemical processing industry, apply it to individual cells. They're a chemical processing unit in themselves. And so we're interested in their basic phenomena and how they might be describable and modelable and therefore predictable in some way in their behavior under certain conditions, including therapeutic treatments possibly. So that's been a major goal for me for many years, and what makes this exciting time for me at least, where we can start to bring the advances in computational modeling and computing power to combine with advances in experimental methodology, namely, for us at least, green fluorescent protein and the genetically encoded markers and the digital camera combined with the computer has allowed us to start to make some of these connections between fundamental theoretical principles and basic cellular processes such as cell migration. So this is my outline for today. The first thing I'd like to do is introduce you to what we are calling a motor clutch model, and I'll explain what that is in a bit. And then I'd like to tell you about 2 different studies that are different but related that both highlight what I think is an important concept I'd like to bring out today is optimality, that in engineered and biological systems, they very often have an optimal operating condition where if you deviate from that one way or the other, it starts to fall apart. So my computer, for example, will work well in a certain temperature range, but when it gets up to 100, 110 or something like that, it's not going to work so well. And when it gets down to 30, 20, 10, 0, 1 degree, I know my iPhone always shuts down after about 15 minutes of playing broomball in January when it's sitting next to the rink. So there's a certain optimal range for things. And I'd like to highlight 2 studies that really make that point, I think. And then at the end, I'll just talk a little bit about a new initiative here that involves Dave Largaspada and myself and others as well who are here and also beyond the University of Minnesota in a Physical Sciences and Oncology Center. So my foray or entrance into the cell migration field, particularly with respect to cancer, was due to being introduced to glioblastoma and its progression grade 4 astrocytoma. And Steve Rosenfeld at the Cleveland Clinic saw some of the work that we're doing and thought that we might be able to apply some of the modeling we're doing to understand the progression of this disease, which hinges quite a bit on cell migration, the ability of the initial lesion cells that are there to migrate out into the surrounding brain parenchyma and eventually take over the whole brain and lead to the demise of the patient almost without exception on a 1 to 2 year time span typically, even with combination of chemotherapy, surgery, and radiation. So this is a real daunting and challenging problem, but it does seem to hinge on the mechanics of cell migration, and at least in part, so that if you could limit cell migration, then you might be able to keep the disease more local, and you might be able to come back with these local treatments like surgery and focused radiation and limit the progression that way. Or maybe even more optimistically would be to tip the balance of the immune cancer cell battle that's going on in favor of the anti-tumoral immune cells that are present in the tumor environment. At any rate, to understand how these cells migrate, we have a number of experimental platforms that we are using and developing. One is very standard for us is now using deformable hydrogels. In this case, you can't even see the hydrogel because it's basically mostly water and it has the optical properties of water, which means it's great for imaging. You can basically image right through it in fluorescence or phase. Here's phase contrast and a standard cell line, the U251 glioblastoma line. But we've, and I'll talk about this towards the end, we also have patient-derived cells that are more low passage and freshly isolated. Also, in parallel with this, there are animal models that we're using, and this is where we're collaborating with Dave's group and using the Sleeping Beauty method to induce gliomas in mouse brains by injection of plasmids that contain oncogenes that will drive tumor initiation and progression. And we can include luciferase and fluorescent tags, for example, actin or other things as well, and be able to then image the cells using confocal microscopy and dissected brains that have been sliced to about 300 microns and imaged on the confocal microscope. And we can zoom in a bit more too and make higher resolution images. These are a little lower res where you see in the lower left the tumor mass, and you can see individual tumor cells that are out into the brain parenchyma and moving dynamically and moving it to varying extents. And this is then gives us the ability to witness the dissemination process that occurs at the periphery of the tumor as it's advancing. We can zoom in a bit more, and we can also tag other things as well, like we can add a tag for blood vessels using isolectin B4 in the red channel. And you can see then here's a host of tumor cells. These were actually added exogenously to a brain slice, so they're the U251 cells fluorescently tagged with actin, GFP, and then the red tag on the blood vessel. And here you can see a capillary in red, and you can actually see now the deformation forces that the cells can exert on their environment in situ in this brain slice preparation. So you start to get a sense that these cells are exerting forces on their environment in order to move through it. And the environment that they're encountering is a complex, potentially 3D environment, although if they're invading along a capillary or blood vessel, that could be quasi 1D. And if that vessel's large, around the circumference of that vessel, could be quasi 2D. Okay, so these cells could encounter 1D, 2D, or 3D environments at various points in the progression. So we'd like to be able to microfabricate different dimensionalities. I showed you a 2D surface earlier, and this is more or less a 3D system here, although you could view it as quasi 1D. So we've created quasi 1D environments by microfabrication, and here you can see on the right a whole host of glioma cells that, if their nuclei tag in blue, and then imaged by phase contrast, and you can see their ability to migrate into these channels and invade to the left and even squeeze their nuclei through the constrictions. The nuclei are much larger at rest, or at least when they're migrating at the wide part of the channel, but they can dynamically squeeze their nuclei, although they pause at those constrictions. And we can create kind of these torture chambers where it makes it hard for cells to get through. We can investigate how the spatial geometry, the confinement, might influence their migration as well. And then lastly, we also can get freshly resected GBM tissue, working with Matt Hunt in the neurosurgery department here, and stain, for example, nuclei and blood vessels, as I showed earlier. And here, just looking at nuclei in the green channel, in the blue channel, but just showing it in grayscale. And you can see, just to show its feasibility, there's no real conclusion to draw here, other than you can see individual nuclei moving about, as well as you can see nuclei that line blood vessels, as you might expect. So we'd like to understand this process of cell migration and how it works. What are the mechanics of it? Because if we could understand that, that might give us some toeholds where we could go in and start to target in ways that would limit the advance of cells through their environment. And so if you look at cells migrating, very often they're extending these dynamic protrusions that you see and then retracting them. So there can be multiple protrusions per cell, and they're fairly dynamic. And so each of these protrusions then can be generate a pulling force in its environment, and it's that force that we'd like to start to understand, so we could understand the balance of forces and how that gets disrupted to allow cells to move. So then the first part of my talk will focus on this motor clutch model, what I'm calling motor clutch, which I'll now explain what that means. So for a buildup over a period of decades, even going back to literature in the 1930s, but best summarized by some reviews in the 80s and 90s, is this idea of a motor clutch mechanism for how cells transmit force to their environment. And the basic idea is that filamentous actin, this rod here shown in the center of the diagram, self-assembles by addition of units at the leading edge, or the plus end, of the actin filaments. And that can essentially, by thermal fluctuations, allow the membrane to ratchet forward as you keep inserting more units. It'll keep pushing the membrane forward, and that creates a protrusion. But at the same time, inside the cell are myosin II molecular motors, and these walk along actin filaments towards their plus end. And if you have oppositely oriented plus ends, then the myosin motors shown on the left here are going to walk towards the plus end, but maybe be pulling on the other side, so you could view them for the moment as anchored. And therefore, they're going to translocate the actin filaments backwards or retrogradely away from the leading edge at some rate. We'll call V filament. And at the same time, then, the third factor in all this is that adhesion bonds can form but also break. And when they form, they could start to come under load and get stretched out. So there's some kind of K on and K off. There's some kind of spring constant that describes the mechanics of that bond. And then the off rate might be potentiated by the stress that that bond is under. And all those forces that are present in that adhesion must balance the forces in the substrate, which may have its own compliance kappa sub at the same time. So the cartoon drawing had been in the literature, but what my student Clarence Chan did was basically start to put these parameters and the mathematical relationships that go with them together to create a motor clutch simulation. So the motor is the myosin II, and the clutch are these adhesion bonds. You could think of the adhesion bonds as being things like integrins or cadherins. There's a whole host of cell adhesion molecules, and I'm going to come back to this a little bit later as to what a potential clutch in glioblastoma might be. And so I won't go through all the equations, but I'll pick a couple key ones. One is the force-velocity relationship for myosin motors, or motors in general. And this is really just saying simply that the more load, and the load is the amount the substrate is stretched, x sub, times its spring constant, which then by Hooke's law gives you a force, a spring force. So the more I stretch a spring, the more force it takes. If you normalize that to Fm, the motor force, you can eventually get to a high enough force in the spring that it equals the motor force, and that ratio becomes 1. So 1 minus that is 0, and the filament doesn't move. That's everything in parentheses there. At the other extreme, if there's no force at all, x sub 0, I let the spring relax, then that ratio is 0, and I have 1 minus 0, so everything in parentheses is 1, meaning that the filament moves at VU, the unloaded velocity. So the more load I put on it, it will start at unloaded velocity, but it'll just slow eventually down to 0 until it's stalled. This is familiar from muscle, and it's sometimes called the Hill equation for muscle, and the literature for that goes back to the 50s. And it's been documented for individual molecular motors, including myosin, kinesin, and dynein. The latter two are microtubule motors. The second key part is that once the bonds come under load, then their off rate starts to increase. We tend to fail the more stress we're under. And that's all this is saying, is that the bigger the load on the clutch, the ith clutch, the faster it's going to fail, and exponentially so, scaled by FB, some bond rupture force. So those are the basic elements of the motor clutch model. And I can pause there if there are questions about this. Otherwise, I'll go ahead and start showing you what the model does and some of the predictions that it makes and how we tested it. Okay, well, feel free to interrupt at any point, and happy to discuss. Basically, there's nothing more complicated in here than just force velocity of muscle, and bonds fail faster when they're under load. That would be the short version of everything I just said. So if you start to just put this together and then use what's called Monte Carlo simulation, where you draw random numbers using MATLAB or any computer language has a random number generator algorithms associated with it, which interestingly came out of the Manhattan Project. So big project yielded this method called Monte Carlo simulation to simulate radioactive decay, which is important for making an atomic bomb. So if we think about, just pause for a second, if we think about cancer moonshot and what that means and what the outcomes of that might be, I think if there was a big investment that was akin to a moonshot or a Manhattan Project, there might be really amazing beneficial things that would come down the road out of that we can't even describe or articulate or even envision right now. But we use that method, as does almost everybody else who does stochastic simulations. And it basically looks like this then. We can simulate different scenarios. If we start in the top, we have depicted the molecular motors pulling the actin to the left. And as they are pulling, though, the bonds here, which are these set of springs here, can engage. And then they'll start to get stretched. And as they stretch, we color code them as a warmer color. So they start out blue, meaning low load, and as they warm up to red, they're under high load. And you'll see then in this top case, those bonds really form and break really quickly. And the reason they break quickly is the substrate spring is very stiff in this case. You see how quickly it changes color without really moving much. And therefore, the bonds load up very quickly, and therefore they fail quickly. And it's very hard to sustain loads in this case. So it's poor force transmission. And you get this kind of frictional slippage. If you watch a marker, this green dot that's placed on the actin move to the left, you can see the retrograde flow occurring in the model. On the bottom, you can also see the retrograde flow, and you can look at other components. And here you get a completely different behavior. This spring, unlike the top spring, is very soft. Now the simulated cell is pulling on a slinky. Okay? And it takes a while, if I pull on a slinky, to get it up to high load. Okay, so I got to keep walking, and it takes time to get there, and I can only walk so fast. Why? Because I'm using myosin motors that can only go so fast. So everything you see in the model is almost replicatable at human scale because it's actually the same elements. There's just more of them, more myosins together, but working against a slinky instead of a car strut or something very stiff like that. But eventually that will get to high load, and the forces must balance, and eventually then the bonds start to come under load, and then they fail. But if you watch this loading cycle as it repeats, you'll notice bonds fail before you get to that high stress state, and every time they do, they let go a little bit of the strain energy that they were trying to build up, which then frustrates the buildup of load and allows it's a relatively inefficient way then to transmit the force because the bonds keep giving up prematurely. And if you notice the middle case here of the 3 green dots, it's moving the slowest to the left of the 3 if you watch this one. Meaning that although there's the same number of motors and same number of clutches and everything, the ability of the myosin to actually move the actin retrogradely is limited compared to the other 2 cases. So this means then correspondingly that force transmission is maximal in this middle case, and that it's relatively low in either the high or the bottom cases because it's relatively inefficient for the reasons I mentioned. And in this case now, you also see this load and fail dynamic, but it happens at a faster frequency than in the bottom case. And so what that means then is that just as the last few bonds are forming in this case is about the time the whole thing is going to fail. And that then now makes most efficient use of the adhesion bonds in order to transmit max force. It has nothing to do with what's going on inside the cell. It's just reading out the mechanics of the environment, and that's it. So it's a pretty simple model, but it now can make some predictions that we can test. And we did that then, and I'll focus now on how this motor clutch model exhibits this optimality with respect to environmental stiffness. So you can see the deformations in the environment if you look at this, and we started out at this point early in the study using embryonic chick forebrain neurons, and here transfected with GFP actin, and you can see the growth cone of the neuron exhibiting these protrusions, these spiky things are known as filopodia, bundles of F-actin. And if you watch this one bead here, which is a red fluorescent bead embedded in the hydrogel, you can see this filopodium pull on its environment. You can see the red bead move in, but you can also see it very abruptly just snap back to some other location. So you see this load and fail dynamic in these movies at sufficiently compliant environments. And here, if you look in this window and make a montage, you can watch that bead over time load and fail. And if you make a chymograph, a space-time plot, and look at this distal reference bead compared to the proximal reporter bead, you see these sawtooth type of patterns with very abrupt failures. And if you film them even faster at 10 hertz, they fail on a milliseconds time scale. So they load up slowly, and then they go boing right back. So it's kind of like plucking a guitar string, if you will. So we can see the load and fail dynamics. And we can measure the retrograde flow rate, here again now looking at GFP actin in a growth cone. And you can see the actin transported towards the middle. This is where it disassembles, and then it diffuses and reloads with ATP back out to the edge of the cell where it can be reassembled into actin filaments. And we can measure then by looking at features as you move from the edge to the center of the growth cone, you can look at those features move in space and time, and you can see these streaks of F-actin density fluctuations moving backwards at measurable rates. And then do that at different stiffnesses, and now you can look and see whether you get a minimum in retrograde flow, as the model would predict, at certain stiffness. So Clarence did that with the growth cones, and it looked pretty good in that at high stiffness, you see high retrograde flow rate, and then as it gets softer, you start to see it drop. It's very sensitive to stiffness in this range here, around 1 kilopascal. Keep in mind, so like muscle tissue stiffness is maybe like 100 kilopascals, much higher than this. So 1 kilopascal is really soft. Glass or plastic is like gigapascal or 10 to the 9th pascals, just for reference. So keep that in mind. 99.9% probably of our in vitro cell biological studies done in petri dishes, which are either glass or plastic, they have Young's modulus that is like 10 to the 9th pascals, but biological tissues have moduli that are more like 10 to the 3rd, 10 to the 4th, 10 to the 5th pascals. So we've been operating, most of our studies have been done way out of bounds from the normal mechanics of the biological environment. And so now we have a system, as do others, we didn't invent it, but it's a relatively simple system to study cells in more realistic mechanical environments. Yeah, so my understanding is non-calcified bone is in the 50 to 100 kilopascal range. And then as it calcifies, it moves up past megapascal. So it would be kind of out here at its softest, and then as it calcifies, it gets higher. Any other questions? I would say in general, it's an open question. I'll just pause here and just say, these are bulk measurements. So you take a bone, you take like a chicken bone, and you go like that, and then you get these. That's how you get the number, okay? Or you take a piece of brain tissue and you put it between two plates, and you go like this or this or this. And so you're measuring it in shearing or torsion or compression or something. You're looking at something on a millimeters to centimeters scale, but cells exist at a 10 to 100 micrometer scale, and their sensing is that local environment. And so the argument we've made is that all biological materials are pretty much composite materials. They have stiff elements embedded in them and then a lot of water and other soft stuff around them. So if you're crawling, if you think of like a chunk of steel wool, it's kind of soft actually if you do this. But if you're a tiny bug that's crawling inside of it using steel, you sense steel, okay? So the steel wool might be a megapascal, but the steel is above a gigapascal. So it's unclear exactly what cells are really experiencing in vivo. But these are just to calibrate what the range is. Okay, and there are other experiments that Clarence did that in interest of time I decided not to go over, but it's published, and I just want to use this as background to set up this idea of optimality. And we tried to go softer, but the cells died, and that was unfortunate. But all the measurements that we were able to make above that indicated that this blue zone is a very abrupt transition, and that if there is a minimum, it will exist around 1 kilopascal or lower for these particular cells. But what we did next was to look at the glioma cells and their behavior, and this was at the suggestion of Steve Rosenfeld I mentioned earlier, and looking at the dynamics of cells on different stiffness environments here, going from 4 to 200, and seeing that in between, at say around 100 kilopascals, seemed to be an optimal behavior, at least with respect to migration. And so we plotted that here. As if you look on the left, is a measure of cell migration speed and other hints of optimality as well, for at least for migration around 100 kilopascals. So now this seems very shifted to the right compared to neurons, which had this optimum around 1 kilopascal. And if you look in the literature, you can find other cell types that have been studied that appear to exhibit optimality as well. Neutrophils around 10 kilopascal and smooth muscle cells around 100 kilopascal. So the question that we started to wonder about is, can our model really explain these different optima? Does it have the ability to be tuned through its parameters to have either 1 or 10 or 100 or whatever optimum we might actually see if we look at different cell types in different conditions? So what we found is actually, yes, the model isn't that complicated. I showed you on one slide what is in it. There's really only, at this motor clutch model, has like 8 parameters in it. So there isn't a huge number of parameters. And we can kind of exhaustively explore it, and we did, all single parameter changes. Change it, put this up, put this down. What happens to the optimum? A very basic thing I just want to point out, and it took us a while to kind of grasp this, but this is fundamental. If you change a motor parameter, like increase the number of motors, you start to create an out-of-balanced system. In other words, what would happen if you just kept increasing number of motors kind of indefinitely? Well, eventually, it would be very hard to sustain force transmission at all because the motors just would overwhelm the clutches. And you would basically have retrograde flow at the unloaded velocity and nearly zero force transmission independent of what the stiffness is. So you lose stiffness sensitivity if you go too far with one parameter one way. Take the motors the other way and dial them down to a very low level, much fewer than the number of clutches, and you have the opposite problem. In this case, there's so many clutches, the motors will pull and then stall. And then every so often a bond would break, but then reform, and this thing would just barely creep along, and the system would always be stalled. So now the unloaded velocity is 0, and force transmission is maximal, and it's, again, independent of stiffness. So in those two extremes, there's no stiffness sensing at all. So if you're going to change one parameter, that will imbalance the system. You have to change a second parameter to counter that, to balance it, so that the system remains in this state where it can sustain forces but also break bonds. And so my student Ben started to look at dual parameter changes, and there's 8 times 8 minus 8 possible changes. But you can do it in silico, and now we're kind of doing screening in silico of our model. We're looking in parameter space of our model in silico and looking at all pairwise combinations that might shift the optimum. To cut a longer story short, I'll just point out that he found a very simple and easy way to shift the optimum is changing motors and clutch number simultaneously to either increase both or decrease both together. This keeps the system balanced such that you have stiffness sensing, and it nicely shifts that optimum to higher stiffness as you increase the number. But you have to increase both together. And he derived a dimensionless number for optimal stiffness that yielded the same results, that n, the number of motors and clutches, shifts the optimal stiffness coordinately with it. Okay, so we were interested whether this might be the reason why glioma cells are different than neurons in terms of their optimal stiffness. So he started to measure the retrograde flow in glioma cells now, and you can see that here. And now we predict that whereas neurons had a minimum in retrograde flow around 1 kilopascal, I showed you, if glioma cells are just simply the same as neurons but have 100 times more motors and clutches, such that their peak in migration would be out at 100 kilopascals instead of 1, then we'd expect to see a 100-fold increase in the optimum for glioma cells, that the minimum in retrograde flow for them should sit out at 100 kilopascals. That's the prediction of the model. So Ben went and measured retrograde flow with U251 GFP actin cells at different stiffnesses and plotted that and superimposed it on the neuron data, and it basically turned out that way, that there was a minimum in the neighborhood of 100 kilopascals shifted to higher stiffness as predicted. And in all those pairwise combinations, there's actually more than just motors and clutches as a way to shift the optimum, but that was an obvious one. And if that were true, then as you shift to higher motors and clutches, you get a higher optimum, you would also increase the forces that you're transmitting in terms of magnitude by 100-fold or so. And so he looked at the traction forces that the glioma cells generate using traction force microscopy, where you look at those bead deformations in the substrate, and you use it to calculate what we're calling here the strain energy. It's the amount of energy that's stored in the substrate as a result of the cell pulling on it. And the glioma cells are able to store more than 100-fold more strain energy than neurons under identical conditions. So this made us think that that is probably what the key difference is between glioma cells and neurons, is simply the number of motors and clutches is the main difference between them. Otherwise, from a motor clutch point of view, they're basically the same. And if that's true, then we should be able to take glioma cells and shift them down if we could simultaneously inhibit motors and clutches together. So that is a doable experiment because blebbistatin is a drug that inhibits myosin 2 motors, and cyclo-RGD is a competitive inhibitor of integrin-mediated adhesion. And we found that both of these would influence the system in that blebbistatin would inhibit retrograde flow, and cyclo-RGD would inhibit adhesion. If you put enough of it, cells round up and lift off. So if you use them at intermediate concentrations and coordinately change them together, you would be shifting motors and clutches coordinately down without completely obliterating the ability of cells to adhere and migrate in their environment. And it's easy for me to say, but Ben spent a year just collecting this data, because if you're going to propose a double drug experiment, which we did, you have to do the single drug controls to go with it. So now that's 3 different conditions for each one, and you have to do that on different stiffnesses as well. And he had 5 different measurables. So this is just now taking that year of his life and just boiling it down to a couple slides, which is always just so sad for graduate students, I know, but hey, I've been there. So, all right. So what it would predict then is that minimum retrograde flow for the glioma, which is out at 100 kilopascals, should start to shift down to a lower stiffness if he double drug treats. And that's what he found, basically. So if he treats with both drugs together, you still have retrograde flow, and it's in the same scale, but now this minimum that sat out here is now shifted to a lower value. And here's the single drug controls as well, just for comparison. And they behave as you'd expect. If you come in with some blevastatin, you can slow down retrograde flow. If you come in with cyclo-RGD, you can speed it up, and that's independent of stiffness. But what's interesting is with a double drug treatment, you can shift the optimum to the left. With this whole time, we've been talking about a motor clutch system, like one protrusion, which we can study. But we know that that protrusion is linked to other protrusions. And they're all pulling on each other. And we've never really gotten, at this point, we had not yet gotten to, well, what about a cell actually moving through its environment, which is the biological and medical thing we really cared about at the outset. So we took to upping the model now to create a simulator of how cells migrate, to integrate the motor clutch, what we'll call modules, together in a force balance and have them duke it out, have them battle each other to figure out where the cell's going to move as a result of them each pulling in their own individual and stochastic way. So here we've got one motor clutch module pointing to the right and one motor clutch module pointing to the left, and they balance each other now, and they each have their own compliance as well. And if we impose that, plus mass balances on the three entities you see depicted in the cartoon, blue is motors, red is actin, and green are adhesion bonds. You could think integrins, for example. And those, if the left one has less integrins, then the right one has more. But they have to add up to a constant, and that's true for all three of those species. So that conserves mass, and forces are balanced, and we can basically chain them up together and have them battle it out, each pulling in their own direction. We can birth in or create new modules or nucleate them, and if they get too small, we quench them and redistribute the remnants to the rest of the cytoplasmic pool, which we also keep track of. And so this is kind of a cartoon schematic, but in real life, when I saw this woman walking down the street in Boston last spring, I thought that she is the cell migration simulator. So what we're shooting for is something like that, a simulator where has anyone played with a flight simulator at all? A couple of heads nodding. Yeah, they're kind of fun. You can crash, it doesn't hurt, you know. And so you have all these controls and knobs you can turn. They have parameters that set the simulator. And the settings are obviously different if you're flying a Cessna than versus a Learjet versus a 747. But they're all airplanes. They just have different parameter sets that make them different. And then you can kind of see how it would behave if you change this knob or that knob. That's basically what a simulator is. So we're planning to do and are doing the same thing, but with cell migration in this case. And so here's what that looks like now. 3 modules pulling on each other stochastically. A 4th module gets nucleated, disrupts the symmetry. A 5th one comes in, one gets too small and it gets quenched, and this guy wanders away. So we can now change the environment of the simulated cell to be at different stiffnesses, like low, medium, and high, and then predict whether there's going to be any interesting behavior as a function of stiffness in the environment. Now, not just for a single protrusion, but for the whole cell. So now you see as you go from low, medium, and high, you go from slow migrating and not very well polarized or spread out to well polarized and spread out and fast migrating in the middle to then again, back to slower migrating, less polarized, less spread at high. This then shows this biphasic behavior, this optimality with respect to stiffness that we had seen in the single clutch modules. So in general, I think of this as like a Goldilocks phenomenon. You know, Goldilocks tried out 3 different beds, Papa Bear, Mama Bear, Baby Bears, and you know, the Papa Bear and the Baby Bear is too stiff and too soft, so Mama Bear's bed is just right, and fell asleep there. So these cells, they don't fall asleep at the intermediate stiffness. They wake up and they energize and they start moving like crazy, which is bad news from a disease progression point of view. So if this is true, then the double drug treatment should move the optimal stiffness for migration from what it was, like around 100 kilopascals, down to a lower optimum. That's the prediction of the model. And so Ben did that as well, measured cell migration, as well as area and aspect ratio, which I'll just skip over, but we're consistent with this prediction as well. And what he found was basically that at lower stiffnesses, the cells seem to pick up steam and a new kind of optimum emerging at lower stiffness and a loss of the optimum that was previously existing at 100 kilopascals, consistent with this coordinate shifting by motors and clutches. And what I think is really interesting about this is it really highlights optimality and how confusing it could be without having a model to contextualize it. And that's illustrated here. If we just looked at the raw data and said, here's two different environments, low and high stiffness, and here's the measurement of random motility as a function of one drug or the other or both. And what happens? Okay, well, first of all, in control, blue, cells migrate fast at 100 but slow at 4. Oh, then you think, oh, they like 100. But why? It's not clear, but that's just a fact. Okay, then you come in with one drug like blebbistatin. Okay, at 4 kilopascals, it has almost no effect. That's here. But added at 100 kilopascals, boom, it just drops way down. Okay, so they're sensitive at 100 but not 4. Why? Cyclo-RGD, same thing. Not sensitive too much, but here quite sensitive. Now take those two drugs, which together suppressed migration here. It seems to recover, actually, but it's still overall down. Take the same two drugs over here, which individually didn't do anything, but together at 100 kilopascals decreased migration. Add them at 4 kilopascals, and you increase migration. So now you have opposite effects of what the double drug treatment does, depending on whether it's on stiff or soft. And this is, I think, conceptually hard to sort out, but it becomes kind of simple if you look at the optimality that's embedded in the model, in the system. So the way to explain it then is the following, is that we know in our model, with respect to stiffness of the environment, there is an optimal stiffness, and that optimum shifts dependent on the number of motors and clutches, such that if you decrease motors and clutches, the optimum shifts to lower stiffness. So the results I just showed you are explained in that way, that at high motors and clutches, the control case, the optimum sits out at a high value, like around 100 kilopascals, and therefore they migrate fast, and these guys migrate slow because they're not at the optimum. But now double drug treat, reduce both motors and clutches, both cases shift down to here now. These guys fall out of the optimum and into the slow migrating zone. The optimum shifts to a lower value, and these now become the new champions and migrate fast. Okay, so if we realize that there's this optimality embedded in the parameter space of the model, it becomes easier to understand what would otherwise be a difficult to interpret set of experimental results. You'd know that they're stiffness sensitive in some way, and somehow these drugs influence it, but it'd be hard to predict what to do with a given patient. So I'd like to now just turn to, that shows the optimality and stiffness that I think we've started to understand the mechanism of it from this motor clutch and cell migration simulator point of view. I'd like to focus now on adhesivity, and then I'll conclude with a brief synopsis of this center that we're now starting. So what is the clutch? I mentioned integrins, but there's other molecules. Integrins, there isn't a lot of collagen in the brain. There is a lot of hyaluronic acid. And CD44 is a cell adhesion molecule that can recognize and bind to hyaluronic acid. So working with Jim McCarthy and John Olfest before he passed away, we started to look at CD44 as a potential clutch in glioblastoma. And it sits in that position where through ERM proteins it can transmit forces to actin potentially while binding hyaluronic acid as the ECM. So it's the same thing, it's just different players, not integrins, but maybe CD44 in some cases. So using the Sleeping Beauty method that I outlined earlier that Dave and John Olfest and others have used, we were able to generate different cohorts based on CD44 background, knocking it out and then knocking it back in and then overexpressing. And what we found then is you can control the CD44 level in these different cases. So by Western blotting, assessing the amount in the knockout in blue, wild type in red, and then the knockout, what's interesting is the wild type overexpression in pink here is higher. What's interesting is that the knockout, when you rescue it, way overshoots the wild type, and it's basically like the wild type plus overexpression. Yep. These are from cells that were isolated out of the mouse brains in this case. So 2 lines from each of those cohorts for a total of 8 lines. And you can see that in vivo, both in humans and in the mouse models, there's an increase in CD44 expression in vivo. So first of all, started to look at survival as a function of CD44. And what was interesting, the wild type in red dies relatively quickly. If you knock out, the knockout in blue lives longer. If you rescue the knockout, it seems to recover it partly, but it doesn't get all the way back to the wild type. And furthermore, if you take wild type and add CD44 to that, they don't do well. They do well. They live longer. So you got these ones. The worst case is the wild type, which is the intermediate CD44 level. If you go above that or below that, the mice live longer. This, again, yeah, this is an induced tumor using this injected plasmid model with a Sleeping Beauty. We're not injecting cells, we're injecting plasmids to induce the tumor. It's pretty reliable, like 80-90% of the mice will develop a tumor, and the time scale of their survival is order of weeks. This is this method. I'm sorry I breezed over it. There's a paper, Wiesner et al., which was in Cancer Research, which Dave and John Olfest were the senior authors on that described the method, and now we're using it in these different CD44 backgrounds. So that made us wonder whether this is, again, it showed optimality with respect to CD44 level, that the worst scenario was intermediate CD44. And when we looked, then we decided to look at human survival as a function of CD44 level. And here, each data point here is one patient using data from Phillips et al. In Cancer Cell 2006. Basically finding then that if you do, it's not shown here, but if you do a linear fit on this data to look at survival as a function of CD44, there's no correlation. It's not statistically significant. So you'd say it's not prognostic of anything based on that. But because we had started to think that the actual behavior might be biphasic with respect to CD44, we then took our linear model and added a quadratic term. So we just had one parameter and now fit that and see if that quadratic term is statistically significant. And it was. And the sign of it was positive, which means it's concave up. It didn't have to be. It could have been concave down. It was concave up, and that's also what we would have predicted. Okay, so now we have a statistically significant biphasic effect when there is not a statistically significant linear effect. If you have a quadratic like this, and then you fit a line to that, it's flat. There's no dependence. Okay, so this is what we were finding with survival of humans as a function of their CD44 level. And one last thing is that there are at least three subtypes, and in the Phillips paper they call them proneural proliferative and mesenchymal, blue, red, and green, which actually work out to be low, medium, and high CD44 level with a fair amount of overlap between them actually, but on average low, medium, and high. And if you look at those three groups, in the blue group, the dependence on CD44 is actually negative, although not statistically significant, borderline. The red group is definitely not statistically significant. There's no correlation. But the high CD44 group, the mesenchymal group, actually had a positive correlation with respect to CD44, meaning that higher levels were actually better outcomes within that group, and that was statistically significant. And if you look at those three groups, blue, red, green, low, medium, and high, and the three dashed lines, they all cross at one point. Where do they cross? They cross where the black quadratic is at a minimum. So everything points to that middle case, the middle CD44 as being the worst outcome. And I'll just mention, we looked at the TCGA and got a similar result, and also found bias in the data in that the surviving patients actually have a broader distribution of CD44 than the patients who've perished. And yet, even with just only analyzing the patients who are deceased, there was a statistically significant and positively signed quadratic coefficient, meaning there's this optimality again. But I'm just kind of skipped that over. So we wondered if that might be either proliferation or migration or both, and we looked at proliferation by bioluminescence, and the rates of increase over the time when it's really taking off were not statistically different among the different cohorts. That would be the slopes of these four lines averaged over a lot of mice with all its variability, but not showing any trend with respect to CD44. And this just shows some of the staining that there isn't gross differences in proliferative or apoptotic cells as a function of the background either. But my student Becky had kind of started to develop this biphasic idea for how survival might depend on CD44 level based on older literature that showed that cell migration is biphasic with respect to adhesivity, that at intermediate adhesion, cells get maximal migration. If you go too high, it's too adhesive, cells get stuck and they can't move. If you go too low, they can't ever get grip in the first place and they also can't move. It's only in between, again, where the adhesions are kind of about balanced right to match the motors, that you could actually move. And this was recapitulated in our cell migration simulator. And so Becky started to take brain slices then and measure the cell migration speeds in these brain slices as a function of the CD44 background to see if there was any pattern to it. And this is some of the data that's shown here. And it looks like the red case is migrating faster than the other three, which is what we would have predicted. And she could quantify that. In fact, it was. So cell migration speed here was statistically different than these other three conditions. And it also had a biphasic effect, and it was inversely correlated with survival. So where you get fast migration is where you get poor survival. Otherwise, they do well. So what we found in this study then was that both mouse and human survival are biphasically dependent on CD44 levels. So if you're at the intermediate CD44, that's the worst case scenario. And that it was not really dependent on proliferation, but it was biphasically dependent on, or correlated with at least, migration. And the biophysical model predicted that. So this highlights again, I think, that the optimality of the system, in this case with respect to CD44 level, is predictive and independent of the biophysics, just from a biomarker point of view. If you're going to start saying, let's look at correlation like high CD44, low CD44, high gene X, low gene X, pick whatever you want. What we're saying is that maybe we got to go a step further. If we don't see a correlation there, it may not be the end of the story. There may be a low, medium, high, either this way or that way, that may be significant, statistically significant and meaningful and predictive. Here we have a single gene, CD44, that has a statistically significant biphasic effect in a cohort of less than 100 patients. So I think this is something that to think about in generally for biomarker discovery, independent of mechanism. I think I'm getting close to the end here, so I'll just spend maybe 1 or 2 slides on this Physical Sciences in Oncology Center and using the cell migration simulator that I showed you already and how it might hopefully be applicable to drive new therapies and be predictive for patients. So I just want to highlight a couple examples of how to think about this and how we're thinking of it at least. So we'd like to know where patients sit in this parameter space to know how their cells are going to migrate. So the fast migration, the orange spot, is bad news from a patient point of view. You don't want to be there. You want to somehow move patients into the white zone, which is a slow migrating state. So if, for example, we parameterize this set of patients in blue in this region of parameter space, that's predicted to be a not very good outcome for them based on cell migration alone. So if we could move them into any white space, that would be good. The closest white space to them is in the southeast direction, this black arrow. That means increasing A1 and decreasing A2, if we had some means to do that by drug or otherwise. You could try to move them in northwest direction, but that's a long way to go. And if you come up short, you might actually move them into a faster migrating state than they started in, and they would actually get worse, possibly. You might exacerbate the progression. Meanwhile, another cohort in red over here might parameterize in this region of parameter space, some of whom are in the orange region, some of whom are already in the white region. So this would suggest to not treat these patients, at least with an anti-migratory strategy, while treating these patients would be beneficial and easiest to accomplish by moving in the northwest direction. Not southeast, the opposite treatment of the blue patients. Without knowing the map, then we don't know whether to inhibit or increase activities that might be relevant to cell migration. And along these lines, I'll just mention then that selingitide, which is basically cyclo-RGD, was used in a clinical trial for glioblastoma and got to phase 3 clinical trial and then failed. And the argument that you could make why it would have failed, and I'm not saying I know why it failed, but this is what we would have predicted going in, is that without stratifying the patients, it's probably going to fail because the patients are going to stratify across that orange region. Some of them will be in the middle, and some will be on either side of the hotspot. Now let's inhibit them. Okay, the guys who are below the already below at the low side of the minimum, if you inhibit them, they won't do better or worse. They'll be unaffected. If you're at the optimum and you inhibit, you can move them out of the optimum, they would benefit. But now take the group that was above average adhesion, partially inhibit, and you might actually move them into the hotspot and make them worse. So now you take all three groups, average them together, you got no effect, got better, got worse. You have a failed clinical trial, and it's phase 3. So I would propose that it might be possible to resurrect selingitide, but stratify the patients according to the target, in this case integrin, and find the patients who are in that, likely to be in that middle group, and then try to target them and see if you can't do better. So that's the plan then, is to try to take cells and tissues from patients, and in complement to the standard histopathology and complement to omic analyses that are emerging, do live cell measurements like I was showing you to extract live living pathology out of patients. Instead of just fixing it right away, let's take more of a transplant model to the operating room and treat that resected tissue as precious and alive and capable of yielding live dynamic information before we throw it away. And so in a 24-hour window, we can parameterize potentially through these mechanical parameters and simulate and predict how patients are going to progress, and possibly longer term, we didn't propose this, but try to find drugs that could target those things and maybe even test them while the patient is recovering from the surgery, and then bring that back to the clinic to come up with diagnosis, prognosis, and treatment. So that's the center, and I'm happy to answer any questions that you might have about any aspect that I talked about today.
So I did want to mention that there was, due to a tragic error, coffee and snacks didn't come this morning, but they will return next week. I wanted to start with one question for Dave. And so you mentioned that the actual stiffness or Young's modulus that cells experience inside a tissue isn't known. But I was sort of wondering whether you could use F-actin retrograde flow as a probe for determining what the stiffness was. But then it occurred to me that the line's not linear. Mm-hmm. So you wouldn't know which side of the minima you were on. Mm-hmm. But if you use the cell as a probe for endogenous stiffness in the tissue, and you could pharmacologically or genetically alter in the number of motors and clutches, and then see how that changed retrograde flow, maybe you could do this. Mm-hmm. Could you use the cell as a probe for the real stiffness that it feels?
Yeah, I hadn't thought of it exactly that way, but that's what I've kind of thought. Is I showed you a movie that had glioma cells going in. They're U251 cells, which we've studied extensively, and I showed you all this U251 results. And then tag them in green, and then send them into tissue. And I showed this movie where they can grab onto and deform their environment. We've kind of pre-calibrated what these green guys can do, and I know there's a whole ensemble of here, but we can find single cells too. We've already calibrated what their max force generation is. So he said that's the force. They're force generators now, which we've pre-calibrated outside the brain slice. Then we let them go into the brain slice and do their work, measure the deformations, and then take the ratio to get spring constant. That's what I was thinking, but it's interesting to think about other ways to do it too.
So this is really cool, and for a non-engineer, you actually made it almost understandable. So, but I was thinking, all of your measurements, I think, were based on stochastic interactions, right? And so the rate of selection in that model is going to be pretty slow, and that might actually mimic progression. But I wonder what happens if you add directionality. So CD44 integrins, all these things tend to cap, right? And so now you've got all of the guys working in the same direction. Mm-hmm. So what happens then? Are your motor clutch models additive, or do they essentially get defeated because now everybody's running in the same direction and they just create slippage?
Yeah. So we haven't added any, there's no bias in how these motors and clutches get distributed and the actin to the different modules. They each get equal share, basically. However, what we've found is that under certain conditions, and I kind of glossed over this, we had to add springs in the center of the cell to avoid it from just running off the stage almost immediately, which is an interesting prediction because basically what happens is if the center of the cell doesn't have its springs and the left and the right are battling, once one side loses and there's no center springs, then it just takes off and it will just spontaneously polarize. And even though they have the same number of clutches, the losing side can never win again. Because every time they get one bond formed, they're going against 50 or 100 on this side. And and the forces balance, so these are under way more load. So then the most likely thing that's going to happen next is that one bond will fail. So we move a little bit further this way. Then another guy tries. He gets mowed down. And it just never resettles. So you can get spontaneous polarization in the model without even adding it. It just happens sometimes. You add the center springs, that gives a little buffer to let the losing side get their act together so they can start to get more clutches in there. And that now it can shift around. But you could then superimpose on the model, if this is what you're saying, Jaime, I hope I get it right, but that you could add like feedback of like there's some polarization signal that biases transport of integrants this way and not that way. That could always be added. My colleague Victor Barocas, who's also part of this PSOC, says, if you can say it, I can model it.
I second. That was a very nice talk. So in terms of your GBM patient cohort, was that, and I'm sorry if I missed this, was that based on gene expression of CD44?
Yeah, I glossed over it. It's mRNA.
It's
yeah.
mRNA.
Yeah.
So do you have any data suggesting that there are actual differences in cell surface expression of CD44 in the tumor cells? And or have you considered maybe that infiltrating stromal cells, non-tumor cells, might be contributing to that as well?
Yeah, we've thought about that, and we haven't done it. What we did was we did the easiest thing, was we took published data that was in a database and pulled it, and then just ran the correlations on it.
The CD44 is expressed on a lot of cell types.
Right, yeah, and the immune part of it,
Yeah.
I mean, it could be just as informative as the immune part, too, except I would think if just hypothetically, if the CD44 was really coming from the immune part, and we showed actually in the staining, I should have mentioned this, the immunostaining shows that it's the tumor cells largely that are expressing the high levels of CD44 in the histopathology. But if it was the immune cells and they were anti-tumoral, then I would have expected the opposite, that actually the intermediate CD44 would have been the best outcome for the mice and humans. So that's why I predicated it to say if they were anti-tumoral. So that's another thing, I didn't get to this, but in this PSOC framework is we intend not to only parameterize the cancer cells themselves, but also immune cells, both pro and anti-tumoral. So I picture there's 3 species that are relevant to parameterize, and you want to make sure you come down as hard as you can on the cancer cells and the pro-tumoral immune cells, but spare the anti-tumoral immune cells that might actually suppress it. And if you could just tip that balance by drawing a line in parameter space and saying, you two are over here, and this, if you have 3 points, you can always draw a line between 2 of them and the other and say, we're going to hit these guys, but spare these guys. That's the bigger picture. But it says a point well taken, we don't know, but it's worth looking at.
One more question from Doug.
David, can you spend a little more time on how you can calculate Young's modulus in human tissue specimens? And the reason I bring it up, outside of brain, if you think about other cancers, migration out of the site of the origin is most relevant clinically. And as somebody who deals in breast cancer, we know we see breast cancers that rise in all sorts of different stromas. So you get a breast cancer in completely fatty breast tissue, you get a breast cancer in solid chunks of collagen, heterogeneously dense tissue on mammography. We understand that the environment of when breast cancers originate, there's a risk to have heterogeneously dense breast tissue because you get more cancer. But I'm not aware that anybody's ever thought about, are these higher risk tumors, or where all of these different tissue stiffnesses are on the scale of the map that you drew. So at any rate, that's why the original question, how it would be easy to do this in breast cancer. I know your grant is not around breast cancer because we get tons of tissue to be able to measure Young's modulus if you can do it.
So the center's focused mostly on GBM and pancreatic ductal adenocarcinoma, but there are smaller components that we did put in there that pertain to medulloblastoma that Dave and I are planning to work on, and then also breast cancer. So if I go back to, I didn't get to cover this, but I'll just show you that Paolo Provenzano, who's the leader of Project 2 and focus on the extracellular environment, has found that these signatures of how the stromal environment, namely collagen, is aligned, influences the ability to invade, and that the mechanics of this then would be the relevant mechanics to look at. So he is developing experimental systems to visualize, this is his work now, Provenzano, visualize breast carcinoma cells in different extensive alignment of the fibers to try to look at that spectrum from a fairly benign to more aggressive scenarios. And then in these systems, if we can get the structures right, we can start to look at what are the mechanics of from one cell's point of view in that environment, what are they looking at? Because if you take that collagen gel and just do this, you're going to get the same numbers that everyone else got, and it won't necessarily be informative of what cell actually senses in its environment. So I don't have a great plan for how we measure that, but I think we got to at least start to get down to this level and think about maybe the cells themselves as probes of their environment. If we can see the deformations, you can see it moving, we can measure it, we know how much force they can generate, that can start to get us a handle on what those stiffnesses are. And can I mention one more thing? This center has attached to it a minimum amount of money that has to be spent on pilot projects. And so we intend to open this up to the broader community here to find other people who might have ideas about how they could connect their work to this center. And so look for that in the next month or two, an announcement, and we'll be looking at proposals that will just allow those types of projects to get going too. I meant to say that.
One other announcement before you guys head out. Just a reminder that registration is open for the annual symposium. Head over to our website. Registration is required this year. It's free as usual, but just head to the website and submit your registration. It takes about 5 seconds. Thanks.